Hello and welcome to Sam Biology class. Friends, today we shall discuss about stomatal movement. Stomatal movement refers to the opening and closing of stoma. There are different types of stomatal movement. They are photoactive movement, scotoactive movement and hydroactive movement. In case of photoactive movement, light controls the opening and closing of stoma directly or indirectly. It occurs due to the hydrolysis of starch in the guard cells or due to biosynthesis of organic molecules or due to ion uptake by the guard cells. Scotoactive movement is seen in case of succulents. In these plants, for example Opuntia, the stoma remain closed during daytime and open during night time. This is to reduce the water loss. In this case of stomatal movement, the opening and closing of stoma is controlled by organic acid metabolism. Third type of movement is hydroactive movement. When the stoma remains open, the epidermal cells lose water. During midday, because of the greater rate of transpiration, huge amount of water is lost into the atmosphere through stoma. In order to restore the water level of the plant, the stoma remains closed. So, in case of hydroactive movement, the opening and closing of stoma is controlled by water. Now we shall see why the stoma open. This is the figure of a closed stoma and that is the figure of a open stoma. So in this closed stoma, these are the two guard cells and these are the surrounding subsidiary cells. So prior to the opening of stoma, there is ion uptake by the guard cells. In addition to this, there is biosynthesis of organic molecules inside it. So the osmotic potential inside guard cell decrease. Therefore, water potential also decrease. In order to equilibrate, water molecules move from surrounding subsidiary cells into the guard cells. As a result, the volume of the guard cells increases due to the elastic nature of their cell wall. Guard cells can reversibly increase their volume by 40 to 100% depending on the species. It is due to the differential thickening of their cell wall. Such an increase in the volume of guard cells force them to curve outward, thereby creating this opening which is known as stomatal aperture. So, when the osmotic potential of guard cells decrease, in that case, water molecules diffuse into the guard cells from the surrounding cells. So, the guard cells become turgid, thereby causing the opening of stoma. Mechanism of stomatal movement. Several hypotheses have been proposed to explain the mechanism of stomatal movement. First hypothesis is photosynthetic mechanism of stomatal opening, which was proposed by von Mohr in 1856. According to this hypothesis, because the guard cells contain chloroplast, so they produce osmotically active photosynthetes by the process of photosynthesis. So the guard cells become turgid and that causes the opening of stoma. Second hypothesis is starch sugar hypothesis. According to this hypothesis, the stomatal movement is regulated by the interconversion between starch and sugar. This hypothesis was proposed by Lloyd and Loftfield and have been modified by Sayer and Stewart. Lloyd and Loftfield, they observed that The starch content of guard cells decrease during daytime when the stoma remain open, and it increase during the night time when the stoma remain closed. Sayer observed that the pH of guard cells increase during daytime and decrease during night time. According to Sayer, because the pH remain high during daytime, so starch is broken down into glucose 1-phosphate in the presence of enzyme phosphorylase. So the breakdown of starch into glucose 1-phosphate decrease the osmotic potential of guard cells, so water molecules diffuse into it and the guard cells become turgid that causes the opening of stoma. Whereas reverse reaction takes place during dark when the pH remains low, and this reverse reaction causes the closing of stoma. The scheme of Sayer was criticized by Stewart. According to Stewart, unless the glucose 1-phosphate is broken down into glucose and inorganic phosphate, no appreciable change in the osmotic potential of guard cells will occur. So he proposed a multi-enzyme system. According to this system, starch is broken down into glucose 1-phosphate at high pH in the presence of enzyme phosphorylase. In the next step, glucose 1-phosphate will be converted into glucose 6-phosphate in the presence of enzyme phosphoglucomutase, and in the third step, glucose 6-phosphate will be hydrolyzed into glucose and inorganic phosphate in the presence of enzyme phosphatase. So the hydrolysis of glucose 6-phosphate into glucose and inorganic phosphate significantly decrease the osmotic potential of guard cells, so water molecules diffuse into it and the guard cells become turgid that cause the opening of stoma, whereas reverse reaction takes place during night time. So during night time, glucose is converted into glucose 1-phosphate in the presence of enzyme hexokinase. In the second reaction step, glucose 1-phosphate molecules are polymerized into starch at low pH in the presence of the same enzyme. This causes the closing of stoma. This is about starch sugar hypothesis. Modern concept of stomatal movement. Friends, this figure shows various metabolic pathways that is associated with stomatal movement in the guard cells. This is the vacuole and this is the chloroplast, and this membrane is the plasma membrane of the guard cells. So when stomata are open, potassium ion concentration in the guard cells increases several fold. This change in the concentration of potassium ion are balanced by chloride ion and malate ion. In the genus Allium, potassium ions are balanced by chloride ion, whereas in most of the other species, it is balanced by varying amount of chloride ion and malate ion. The chloride ion and the potassium ion are taken up into the guard cells by secondary transport mechanism. In the plasma membrane of guard cells, there are proton pumps. This is a proton pump which is present in the plasma membrane of the guard cell. And this proton pump is activated by blue light. The proton pump is a H+ ATPase. It is an active transport system. It transports H+ ion to the outside of the guard cells. As a result, the concentration of H+ ion increases outside and decreases inside guard cells. This generates electrochemical potential gradient for H plus ion. The electrical component of this electrochemical potential gradient generate the driving force for the passive uptake of potassium ion into the guard cells through voltage regulated potassium channels. Chloride ions are also taken up into the guard cells through anion channels. In addition to this, malate is also synthesized. So inside the guard cell, there is accumulation of potassium ion, chloride ion and malate ion inside the vacuole. So the osmotic potential of the guard cells decrease. So water potential of the guard cells also decrease. In order to equilibrate, because there is a gradient of water potential, because water potential is less inside guard cell and more on the outside of the guard cells, so water molecule diffuse into the guard cells. As a result of diffusion of water molecule into the guard cells, the volume of guard cells increase. As a result, they become turgid, and at last, it is due to the turgidity of the guard cells, the stoma open. But during the closure of stomata, reverse reactions takes place. That means potassium ion and chloride ion, they move out, and malate concentration also decrease. Probably malate is converted into pyruvate. So the osmotic potential of the guard cells increase. As a result, water molecules move out of the guard cells. So the guard cells become flaccid and the stoma gets closed. So the blue light activation of proton pump play a key role in the osmoregulation of guard cells during light regulated stomatal movement. Already we know about starch sugar hypothesis. According to this hypothesis, the starch content of guard cells decrease during daytime. This is due to the hydrolysis of starch into sugars. So the osmotic potential of guard cells decrease during daytime. So the water potential also decrease. In order to equilibrate, water molecules diffuse from surrounding cells into the guard cells. So the guard cells become turgid and the stoma open. But during night time, the starch content of guard cells increase and the sugar content decrease. This is due to the synthesis of starch. So the osmotic potential of the guard cells increase. As a result, water molecule diffuse out into the surrounding cells. As a result, the guard cells become flaccid and the stoma close. But with the discovery of major role of potassium ion and counter ion in osmoregulation of guard cells, the starch sugar hypothesis is no longer considered as important. Studies have shown that there is another major osmoregulatory phase of guard cells in which sucrose is the osmotically active solute. The potassium ion concentration of guard cells increase in parallel with the early morning opening. The concentration of potassium ion in guard cells decrease in early afternoon. The sucrose concentration of guard cells, which increase slowly during morning, become the osmotically active solutes after the efflux of potassium ion. In the evening, when the sucrose concentration of guard cells decrease, the stoma gets closed. These osmoregulatory features indicate that potassium ion uptake is primarily associated with stomatal opening, but a decrease in the sucrose concentration of guard cells is associated with stomatal closure. So there are four different metabolic pathways operate in the guard cells that can supply osmotically active solute. First pathway is the uptake of potassium ion and chloride ion along with the coupled synthesis of malate in the guard cells. Second pathway is the production of sucrose from the hydrolysis of starch. Third pathway is the production of sucrose in the guard cells by photosynthesis. And the final pathway is the uptake of apoplastic sucrose generated by mesophyll photosynthesis. This pathway is the Calvin cycle, which provide dihydroxyacetone phosphate. The dihydroxyacetone phosphate move out of the chloroplast of the guard cells, and in the cytoplasm it is converted into sucrose. Finally, this pathway shows the apoplastic transport of sucrose, which is generated by mesophyll photosynthesis. Depending on the environmental condition, one or several of these pathways can be activated. So friends, this is all about stomatal movement and its mechanism. For more updates, please subscribe my YouTube channel, Sam Biology Class, and also please share your comments. Thank you very much.